Hello to our friends joining us via recording. We are getting ready to launch into lesson number 5 about the way we build proteins, their different levels of structure. We're trying to keep the atmosphere a little bit light, so right now we're sending in emojis and GIFs. Jessica looks pretty excited, giddy like a child. I got a few thumbs up, love it. Got some smileys. Hayden's not loving it. I'm sorry, Hayden. I'll try to keep it alive. Lisa's feeling spiffy. I like it. So hopefully during our class time today, we can learn a lot together. You're going to be doing a really interesting application activity related to protein structure. It's about a disease called Creutzfeldt-Jakob's disease. So we will take some time at the end of class to briefly mention about that, but that'll be the disease that we're learning about here with lesson number 5. Lisbeth, I'm sorry that your thumb's down. That's okay. It doesn't every day doesn't have to be a good day. I'm hoping I can maybe get that thumbs up for you. So that's the goal. All right, so let's dive in to lesson number 5. Lesson number 5 is all about the basics of proteins. So when we talk about proteins, we're talking about something in your cell that's like 30% of what's there. So it's a big chunk of what's inside your cell. Everything else are things like, things like lipids or carbohydrates, but we have a water. We have a lot of protein in our cell though. The proteins that are in our cells are found in different places. So some of those proteins are inside the membrane of the cell. When they're inside the membrane of the cell, they have 2 jobs actually. One of those jobs is to help cells to talk to their neighbors. So we use proteins to communicate. The other reason we have proteins in the membranes of our cells is to help us do all that stuff we talked about last unit when we talked about facilitated diffusion or active transport. We need to use proteins to get things across the membrane. So some of that 30% of the cell being proteins is stuff for communication or stuff for transport. Some of those proteins are inside the organelles of our cells. And when we're talking about proteins like that, they're going to be functioning as enzymes. So their job is to get stuff done, like we talked about in unit number 1. We'll talk about several enzymes, especially next week, that their job is to make our DNA or work on our DNA, help us express it. So some of those proteins found inside organelles, helping them to do their job. Others of them floating around in the cytosol, doing things like our cell's structure, helping our cell to do some of its other enzymatic functions. So all of these things, roles of proteins inside our cell. When we talk about proteins, proteins are made out of a monomer or a subunit thing called an amino acid. So amino acids, like you can see on the screen here, they get their name because of the 2 parts that they have on them. Over here on the left, we have the amine group. So the amine group has an amine group in it. So this is a nitrogen and 2 hydrogens. Amino in its name from the amine group, acid in its name from the carboxylic acid group, which is what we see over here. Now, I promise I'm never going to make you draw the chemical structure of an amino acid, but I am pointing out the amine group and the acid group for you because in just a moment, we're going to talk about the way that we build proteins, and I have to steal things from the amine group and from the acid group to do that. When we talk about the way that proteins are built, everyone needs to underline, highlight, star this term here, peptide bonds. Peptide bonds are the special kind of chemical bond that we only find inside proteins. We use peptide bonds to connect together amino acids. So if I ever tell you on a quiz question or a test question, hey, we've got a compound that has some peptide bonds in it, immediately you should be thinking, okay, we're talking about a protein. So proteins are peptide bonds are the special kind of bond that we find inside proteins, connecting the amino acids to each other. The way that I build peptide bonds is using something called dehydration synthesis. Dehydration synthesis. Hey, if you are dehydrated, what's going on with you? Dehydrated is a big, long word. How might you describe it to my 5-year-old? If you're telling her, oh, you're dehydrated, here's what's going on. Yeah, I like it. Hayden is going real, right? He's saying, hey, you'd be thirsty. Absolutely. Jessica mentioned we'd be low on water. Totally true. When we dehydrate something, like Victor mentioned, we pull the water out of it. So when I'm looking at using something called dehydration synthesis, what this big long name means is I'm taking out water, that's dehydration, and I'm using that to build something, synthesis. Dehydration synthesis. I steal water from an amino acid to build a peptide bond. So this is where that chemistry that I mentioned for you comes into play. If I'm going to steal water from things, water has that formula of H2O, 2 hydrogens, 1 oxygen. If I'm going to build that by stealing things, I need to have both of those things. So the acid group of one amino acid, I will take the OH from. The H from my amine group on another amino acid, I will take those things away. When I steal those things, I build water, but I also build a peptide bond. So peptide bonds connect together 2 amino acids. The way that I do it is by stealing water, dehydration synthesis, to build these peptide bonds. We care about peptide bonds and dehydration synthesis because this is what builds the most basic level of protein structure. So the most basic level of protein structure is called primary structure. Primary structure is the order of the amino acids that are inside a protein. Fun fact about amino acids, we only have 20 of them that are building blocks that we use to build the millions, literally, of proteins inside our body. So the primary sequence of all of these proteins in our body that are so different comes down to the same 20 building blocks, just the way that we put them together. So primary structure tells me what order did I put those amino acids in. Notice I've got one amino acid right here, my blue dot. It's connected to another amino acid here in red using that peptide bond. This amino acid, though, also made a peptide bond with its neighbor over here. And we made another peptide bond with the neighbor over here. So when we're talking about the primary structure of a protein, a lot of times we talk about it like beads on a string. So the order that those amino acids go in, the way they're connected with those peptide bonds, that's the primary structure of the protein. I want to pause for a moment, give us a chance to ask questions or to give me a thumbs up in the chat if you're tracking with me. I see that Lisa already helped Alyssa, but Alyssa asked an important question. We steal parts from our amino acids to make the peptide bonds. Absolutely. We steal our oxygen and hydrogen from one, and we steal our other hydrogen from another. Yeah, so Hayden asked a good question. Hayden asked, where does the sequence of primary structure come from? In other words, how do I figure out what order my amino acids are supposed to be in? That is actually the topic of class next week. So the way we figure out the primary structure of a protein is we look back at the DNA inside your cell. So your DNA, the information molecule, tells me exactly what order my amino acids should go in. So we'll talk all about that next week. Coming soon next week. All right. So far we've talked about how our amino acids have an amine group and an acid group. And we've talked about how we can use those to form peptide bonds to help us build the primary structure, the order of our amino acids. But the other thing we can use the amine group and the acid group to do is we can use them to build things called alpha helices or beta pleated sheets, or often I'll just call them beta sheets. When we talk about building these things, these are what we call the secondary structure of a protein. So the primary structure of a protein is the order that the amino acids are in. The secondary structure of the protein is when amino acids that are not right next door to each other start to interact. So I have one amino acid up here that's interacting with one that's way over here. Or I have one amino acid again up here that's interacting with another one down here. When I have amino acids interacting with others that are far away from them, but we're still using the amine group, we're still using the acid group, that leads me to build these secondary structures. So think about a secondary structure, the alpha helix. Think about that like those curly ribbons you can put on a birthday present. They spiral back and forth in a helix formation. When we think about beta pleated sheets, I kind of like to think of these like an accordion. So a stretched out accordion, I should say. So alpha helices, always this 3D kind of curling thing. Beta sheets are actually flat and they go back and forth and back and forth. Again, this secondary structure is because the amine groups and the acid groups are interacting with each other. But here's what's different about this interaction compared to what we talked about before. Before we talked about building peptide bonds, where we stole the water to get things to connect to each other. When we're talking about the secondary structure, we're not forming peptide bonds. Excuse me. We are forming the kind of bond that goes between a positive dipole and a negative dipole. A positive dipole and a negative dipole. What was the name of the chemical bond that forms between that positive and negative dipole? Do we remember which kind that was? Yeah, so we've bounced around a little bit. Most of us landed on hydrogen bonds. Hydrogen bonds are the kind of bond that forms between two dipoles. So if I have a partial positive and a partial negative that are attracted to each other, that's a hydrogen bond. Some of us mentioned those polar covalent bonds. Polar covalent bonds are at play in this, but remember that polar covalent bonds are why I build dipoles. So polar covalent bonds means I'm not sharing equally. So if my nitrogen is seeing those electrons more often than my hydrogen is, hydrogen gets a dipole. And over here, if my oxygen is seeing the electrons more than my carbon is, my oxygen gets a negative dipole. So I use polar covalent bonds to make dipoles. Once I have dipoles and they start to interact with each other, that's called a hydrogen bond. So building my secondary structures, building my alpha helix or my beta sheets, that's the work of hydrogen bonds. Building my primary structure, connecting my amino acids in order, that's the work of peptide bonds. When we look at proteins, they're never just going to be one little curlicue. Proteins always are more than just one thing. So when I'm looking at this protein we see on the screen right here, this protein has alpha helices and beta sheets. Can we tell from looking at this protein which of my colors represents an alpha helix? Is it the blue or the orange that would represent an alpha helix? What do we think? Exactly. Yep, it's that orange-yellow color where we see things kind of spiraling around. So my alpha helices in this protein, where distant amino acids are interacting with each other to form the spiral, we see that in orange, or kind of the orange-yellow. When we talk about those flat beta sheets, we can see those in blue. So notice those flat beta sheets that we see here. I do want to point out, when we look at this protein, you can see some places that are still gray. So notice these gray things that we see here. The gray that we're seeing here is areas where I don't have secondary structure. So not all parts of the protein are going to be an alpha helix or a beta sheet. Some of them are just going to be flat. They're just going to be a line. That's okay. That's the way that the protein works. But when we're talking about having an amino acid chain with lots of curlicues and lots of flat places, it never stays just flat on your bench in front of you. It always folds up into a 3D shape like we see here. So we need to talk about now how we ended up with a 3D shape. We started with some curlicues, we started with some zigzags. How did we get here? Well, we have to go back to our amino acids to explain how we ended up there. When you look at an amino acid, we have already talked about the amine group. We've already talked about the acid group. What we have not talked about is this R group in the middle. So this R group in the middle is something called a functional group. And when we talk about the functional group of an amino acid, this is the part of the amino acid that determines the biochemical properties of the amino acid. Now that's a big fancy word right there, biochemical properties. All it means is, do you or don't you play nice with water? Do you or don't you want to form a bond with me? When we talk about amino acids and we talk about their biochemical properties, you should think about this as their personality types. So in people, we say, oh, you're an introvert or you're an extrovert. I can't remember what the other ones are. That's the big one that I know. You're a thinker or a feeler, you know, those kinds of things. Amino acids aren't thinkers or feelers. They aren't extroverts or introverts. But what they are is nonpolar, polar, or charged. So when I say that some of them are charged, I mean that some of them are positive, some of them are negative. Then we also have some that are nonpolar and some that are polar. So when we're looking at our amino acids, all of them care about their functional group. Their functional group is their personality type, and that's going to tell us or tell them how they can react, what they can do. Okay, check this out. I promise you, I will never ask you to identify an amino acid based on its structure. I promise I'm never going to give you a picture of phenylalanine and make sure you know what it is. I don't care about that. Honestly, I don't even care about you memorizing which amino acids fall into each of these groups. But what I do care about is that you're able to use the information when I tell you that glycine is a nonpolar amino acid. We better know why that matters. We better know what that means. So help me out. When we were in class, I think it was last week or the week before, we made a list of all the things that nonpolar means. Can you tell me in the chat, what are some things that you can learn about something if I tell you that it's nonpolar? If I say that all of these are nonpolar, what can you predict about these amino acids? What does that word mean to us? Yeah, so lots of us are remembering that that means that we don't play nice with water. We're hydrophobic. If we are nonpolar, as Caitlin mentioned, and I think I saw someone else earlier had said, we share our electrons equally, which means we don't have dipoles, which means we don't play nice with water because water has those dipoles. Yeah, we also, Cassidy mentioned the non-polar covalent bonds. Yeah, so all of these amino acids, they share their electrons equally. We have no dipoles, no areas where we're partially positive or partially negative. We're completely flat. But the big one, which is what we all started with, was the fact that things that are nonpolar are hydrophobic. They don't play nice with water. That's the big key with these amino acids being nonpolar. These amino acids that are nonpolar do not want to be by water. So when a protein's deciding how it's going to squish into a 3D shape, It's going to make sure that these nonpolar amino acids are not touching water. So one personality type, nonpolar. Another personality type that we see here are the polar amino acids. Everything we said for nonpolar is now the opposite with polar, right? So polar amino acids do like to be around water. Polar amino acids do have dipoles. Polar amino acids are going to act the polar opposite of nonpolar amino acids. You like my cheesy joke there? So my polar amino acids, we're typically going to find those around water in places where they might bump into other polar things. The other type of amino acid that we have in our proteins are ones that have a full negative or a full positive charge. With these amino acids with a full negative or a full positive charge, these kinds of amino acids are actually able to interact with each other and form a special kind of chemical bond with one another. Oh, I think somebody accidentally has their mic on. Make sure we're muted, please. Does anyone remember from our discussion last time, what is the name of a chemical bond that forms between something that has a completely negative charge and something that has a completely positive charge? What did I call that kind of bond where you're negative, I'm positive, we get attracted to each other to build our bond because of the negative and the positive? Exactly. Yep, several of us chiming into the chat. We form ionic bonds. So when we talk about amino acids that have a negative charge or a positive charge, we're talking about amino acids that can make ionic bonds with each other. So we'll see a lysine across from a glutamate, a negative and a positive across from each other, and they'll form a nice little ionic bond and connect with one another. Or we'll see an aspartate interacting with an arginine. They'll form a nice little ionic bond and interact with each other. Things that form ionic bonds, they also don't mind being next to water, because remember that these kinds of things with ionic bonds, those bonds dissolve in water. So these amino acids could be next to water as well. They're not afraid of it. They've got a fully negative or a fully positive charge. It's just those nonpolar ones that don't want to be next to the water. So let's take these personality types and explore how that can contribute to my 3D shape. So explore what in proteins we call the tertiary structure. So the tertiary structure of a protein is the way I go from being flat to having a 3D shape. When I talk about that tertiary structure, it all comes down to the functional groups. I want you to make sure to underline, highlight, star in your notes that the functional groups are where the tertiary structure comes from. It's all about those personality types. That's where 3D shape comes from. There are many different ways that the personality types can interact with each other, leading to a 3D shape. So let's go through and talk about these things. I want to point out first that every time we talk about some kind of bond or interaction that things are doing with each other, recognize that we're talking about amino acids that are really far away from each other. Peptide bonds, we were neighbors. We were right next to each other. Those hydrogen bonds, to make my curlicue, we were kind of close to each other. When we're talking about tertiary structure, we're very far away and we're still interacting with each other. So one of the ways that two very far away amino acids can interact with each other is forming hydrogen bonds. Now, remember when we talked about hydrogen bonds, we talked about those earlier today. They're an interaction or an attraction from a negative dipole and a positive dipole. That's a hydrogen bond. Hydrogen bonds, I put them in quotes here because remember, we do not share any electrons. Hydrogen bonds are not particularly strong unless you have a whole bunch of them, and that'll help to stabilize things. So one way that my polar amino acids can interact with each other, things that have a partial positive or a partial negative, they can interact with each other using these things called hydrogen bonds. Another way that amino acids can interact with each other is with a thing called hydrophobic attractions. Hydrophobic attractions are based on the idea that we all hate the same thing, or we're all, in the case of this word, afraid of the same thing. So let's group together and stay safe. I really like to use the analogy with hydrophobic attractions of football teams. So we just had the Super Bowl. Full disclosure, Dr. Ollis did not watch the Super Bowl. I had no dog in that race, as they say. Dr. Ollis is a tried and true Broncos fan. I love me some Broncos, even though they've been bad lately. So I love the Broncos. If you are a Broncos fan, typically you don't like the Raiders. It's just, I don't know where that rivalry came from, but I grew up on it. So I still don't really like the Raiders. So when the Broncos get eliminated from the wild card or, from the playoffs, okay, I can't root for the Broncos anymore, but I definitely am not going to root for the Raiders. So maybe the next week, the Raiders are playing the Chiefs. I'm going to root for the Chiefs because I don't want the Raiders to win. Or the next week, the Raiders are playing the Ravens. I'm going to root for the Ravens. I may not be the same as the people that are rooting for those teams, but we all don't like the Raiders the same. So because of that, we're unified. Because of that, we come together. That's the way that hydrophobic attractions work. Both of these amino acids are nonpolar. Both of them don't like water. We'll squeeze together. If we squeeze together, we're going to push that water out. Now I've got this nice little pocket in here where I don't have any water and all of my nonpolar amino acids are happy. Yeah, Jessica's also a Broncos fan. Go Broncos. One of these years, they'll get back to their glory days. It's been a while. And Lisa mentioned that the Bills hate the Dolphins. I like it. Yeah, there's probably some very particular rivalries that are going on in all kinds of sports. You all know that I came from Oklahoma, so OU OSU is a big rivalry as well. So just think about sports rivalries, and that might be a good way to think about hydrophobic attractions. We may not all be the same. We may not agree on everything, but we do agree that we don't like water, and that's enough to bring us together. Alyssa asked a quick question, a clarifying question about our hydrogen bonds. Do we remember what personality type we mentioned would help us to form these hydrogen bonds? What personality type would be able to do this? Anyone remember? I'll phrase the question another way. If I'm going to be able to build a hydrogen bond like this, I need to have a dipole. So which kind of amino acids have dipoles? Yeah, exactly. Yep. So polar amino acids, ones that either have a positive dipole or a negative dipole, that's going to be the ones that can form hydrogen bonds. Ones that are nonpolar are going to be the ones that form these hydrophobic attractions. Another way that we can put together our amino acids is we can form what are called disulfide bonds. Disulfide bonds are when the sulfur in one amino acid interacts with the sulfur in another amino acid. Since we're interacting two of the same thing in the amino acid chain, we're going to share our electrons equally, which means we're going to make a nonpolar covalent bond. When I talk about a nonpolar covalent bond, is that strong or weak? Nonpolar covalent. Is that a strong bond or a weak bond? Yeah, several of us chiming in. It is definitely a strong bond. It is the strongest kind. Sharing bonds are always strong, but equal sharing bonds are the strongest. So when we talk about how we build the 3D shape of a protein, these disulfide bonds that's like top notch. If you can build a disulfide bond, that's perfect. That's what you want to build because that's the strongest. It'll hold our proteins together. One other kind of bond, and we kind of already talked a little bit about this, the other kind of bond that I can make between amino acids is an ionic bond. So if you've got a positive and I've got a negative, we can form an ionic bond as well. Hey, if ionic bonds aren't around water, they're actually relatively strong because you're fully negative and I'm fully positive. We like to be close to each other, but we get into trouble when we start putting that water around, right? Because in ionic bonds, they dissolve in water. So if we were to put some water next to these guys, or if we were to put some other positives or negatives, this might go away. Same deal up here with my hydrogen bonds. Hydrophobic attractions, we're always going to try to squeeze together to keep the water out. And if we're sharing our electrons equally, you better believe we're staying stuck together. So the reason I go from these alpha helices and beta sheets that are by themselves to the overall 3D shape I see here, that's because of those personality types. That's because of the functional groups. Let me pause for a moment and give us a chance to ask any questions or to make any statements in the chat that we want clarifying on. Or if you're feeling good, shoot me an emoji. I'm going to send you all an emoji while I wait. Here's Dr. Wallace's emoji for you. I do love me some penguins. So Max asked the question, they group together based on their personality types. That's correct. So different personality types squeeze close together because they're going to form some kind of bond or because they all hate water. And the personality types that we're talking about, the way we joke about it, is based on their chemical properties. That is correct. Yep. So we group the nonpolar ones as a group into that personality type, and the polar ones into their own personality type. So absolutely correct, Max. That is accurate. And then Alyssa asked a clarifying question about hydrophobic versus ionic. Yeah, so hydrophobic attractions, it's not so much that, like you said, it's not that we're being pulled together. In an ionic bond, we're being pulled together because you're negative and I'm positive. We want to be close together. In a hydrophobic attraction, it's not that I necessarily am directly being pulled to you, but it's that when we get close together, we realize, oh, like if we stick together, we can both work to keep the water out. So yeah, kind of a good way to think about it. Ionic bonds, they're being pulled together. Hydrophobic attractions, we just kind of exist close to each other because we like that. That's good for us, no water around. Lisa asked a question, does the image show that the bonds are interacting with each other? So these particular types of chemical bonds don't necessarily interact with each other, but we would have a lot of these different bonds going on inside our protein. So this shows us just one little section of a protein with a few little amino acids. We have connections all over the place in a protein. So the bonds themselves don't interact with each other, but our entire chain is going to have interactions like this. Let's see. So Victor asked if the tertiary structure of a protein would have all of these types. It could have all of these types. It may not. Different proteins will have different kinds of bonds because, again, if I have a different primary sequence, so if my order of amino acids is different, then the kind of bonds I can make would be different as well. But they could have all of these types inside of them. Absolutely. Oh, I like it. Alyssa said we could think about hydrophobic attractions being hugging and ionic bonds as kissing. I like that. That's fun. And Samantha asked a clarifying question about disulfide bonds. Let me put that question to the chat. What are some of the things that you all remember about disulfide bonds? What did you catch as the most important things about disulfide bonds? Let me see what we heard as a class. Okay, so Hayden's mentioning these are equal sharing bonds. They're very strong. Yeah, Jessica's mentioning that disulfide bonds only form between two sulfurs. Absolutely. So that's the strongest kind of bond I can use to build my 3D structure in a protein, disulfide bonds. Let me see. Yeah, it's the two sulfurs being stuck together. Absolutely. Yep. So I'm not going to take class time to do this activity with you, but wink, wink, nudge, nudge. This is really great prep for the quiz that we'll take next week. We have talked about primary structure, secondary structure, and tertiary structure of our proteins. When we talk about those different levels of structure, we've now talked about various kinds of bonds that we use to build each of those levels. Some of my levels of structure are built using ionic bonds. Some of them are built using peptide bonds or hydrogen bonds. Please go back in your notes and look over and see what was the specific name of the bonds in primary structure or the specific name for secondary structure. The first thing I want you to make sure that you take away from what we've talked about is the specific name of those bonds that we have inside the different levels of structure. The other thing that's really important for you to take away is which parts of my amino acid are involved in forming these structures. Hey, let's do the last one together, just because I can't not ask you this. What was the part of an amino acid that was most responsible for that tertiary structure, for the 3D shape? Which part of my amino acid is most responsible for that 3D shape? Yeah, several of us putting in the chat. Totally right. It's that functional group or that R group. The personality type dictates my tertiary structure. When we're talking about the other levels of structure, those have nothing to do with the functional group. Those are all about the amine group and the acid group. So please make sure to take some time to go back and reflect on this as you're studying and preparing for our class next week. In addition to helping us know how a protein is going to fold, the other thing that functional groups do for a protein or for an amino acid, is tell them whether or not they play nice with the middle of the plasma membrane. So remember how we started saying that a bunch of proteins live inside the plasma membrane. Some of them, like this one that we see right here, protein number 2, some of them are what we call transmembrane proteins. I put that word in the chat. Transmembrane, which means they start on one side and they go all the way out to the other side. Transmembrane proteins. If I have a part of me that's embedded inside the middle of the plasma membrane, that part is going to have to behave very differently than the parts that are on the outside or the inside of the plasma membrane. And it all comes down to that chemistry stuff that we talked about. So this middle part of my plasma membrane, remind me again, was this part polar or non-polar? What's going on with these tails? Were they polar or non-polar? The middle part of the membrane. Yeah, exactly. These are non-polar. And remember that they're what decides what can and can't get through the membrane. So they're not going to let amino acids be next to them that don't match them. So when I'm looking at this transmembrane protein that goes from one side to the other side, if I want to be in the very middle of the plasma membrane, if I want to be stuck there right next to those tails, I better match those tails. So these blue amino acids here are non-polar amino acids. That's the only personality type that plays nice with the membrane. Same deal with these right here. The part of this protein that's inside the plasma membrane, non-polar amino acids. We see the same thing over here as well. Parts that are embedded in the plasma membrane, nonpolar amino acids. Now, when we start talking about these magenta or purple parts that are either inside the cell or outside the cell, now we're looking at amino acids that are exposed to water. Because remember, when we filled in that activity, there's water outside the cell, there's water inside the cell. We have to be okay with water if we're going to live in those environments. So when we talk about the kinds of amino acids that would be okay with water, what were personality types that might be represented by these purple ones here? Who might be okay with that? Yeah, several of us chiming in with polar. Polar would absolutely be okay with that. Interestingly, so would positive and negative. Remember, things that have a full charge also like water, because water still has a partial positive or a partial negative. So these purple, pink, whatever color we're calling it, these parts are going to be the polar amino acids, the positively or negatively charged ones. The non-polar amino acids are the ones that are going to live inside the plasma membrane, embedded in between those tails because they match their chemistry. How do we feel about this question? Thumbs up, thumbs down, or what other questions do we have about it? Feeling good? Makes sense. Thumbs up. Awesome. Okay. Excuse me. So here's what you can expect on the homework assignment and on the quiz and the exam. I'm going to give you a list of amino acids. I'm going to give you their technical names, but I'm also going to tell you, hey, this one is positive, positively charged, or this one is polar, this one's non-polar, and I'm going to ask you which one of those particular amino acids would be most likely to be inside of here. Remember, it's just like that question we had on the quiz where I gave you a bunch of compounds and I told you, you don't care about the name of the compounds, you just care about their chemistry. It's the same deal with these amino acids. You don't care which amino acid I tell you, you just care, hey, what's your personality type? If I know your personality type, I can predict where I'd find you. That's the goal of this critical thinking question. There is one other level of protein structure. Not all proteins have this, but this extra level of protein structure is called quaternary structure. A protein can only have quaternary structure if it has more than one amino acid chain. So up to this point, we've only been looking at a protein that had one chain of amino acids. But what we see over here on our slide is hemoglobin. And hemoglobin is a protein that has more than one amino acid chain in it. So I'm going to show you kind of a weird picture here. The hemoglobin protein, the oxygen transport protein in your body, is actually made out of 4 different proteins. So I've got these green chains here. They're called alpha chains. Sidebar, this has nothing to do with alpha helices. So this is completely different. This is a chain called an alpha chain. I've got 2 of these proteins inside hemoglobin. I also have 2 proteins that are called beta proteins. Again, not beta sheets, just beta proteins. When I'm building hemoglobin that can function and can take oxygen throughout the body, I need 2 alpha subunits and I need 2 beta subunits. What this weird picture shows you is the way that these subunits interact with each other to build us this really big hemoglobin protein. So scientists went through and they looked at all of the primary structure of hemoglobin. They went through and numbered each and every one of the amino acids in hemoglobin. So notice they went as high as 141. There's probably even more than that. But we've got this amino acid in here, and they looked to see where does this particular amino acid interact with amino acids in the other chain. And so these little dots show me the places where they're forming bonds. This is things like ionic bonds, or things like those hydrogen bonds or disulfide bonds. So they went through and figured out all of the different places that these 4 subunits connect to each other. Long story short, if I'm going to have quaternary structure, I need a lot of chemical bonds to attach these 2 things together. And that's what I see in hemoglobin. So if you have quaternary structure, it took us more than 1 protein chain to build that. It's always more than 1. Now here's something we haven't talked about. I probably should have started here, but I bet you can predict this just from what we know from unit number 1. If a protein is going to do its job, if it's going to be functional, it has to be in the right shape. This is like class number 1 stuff, right? A protein has to be the right shape for it to do the right job. Hey, is it the shape or the function that is the anatomy of the protein? Is the anatomy of a protein its shape or its function? What do we think? Exactly. Yep. So the anatomy of a protein is the shape that it's in, which means that the physiology of a protein is what it does. So when we talk about proteins and their ability to do things in our cell, they have to have the right anatomy to do the right physiology. I have a name for what I call it when a protein permanently loses its shape. It's so important that proteins be in the right shape. I have a name for what I call it when they lose that shape and they can't go back to it. The name of that is denatured, and that's an underline, highlight, star word. A protein is denatured when it's permanently the wrong shape. It can't go back to being the right shape. There are a lot of different things that can denature proteins. Make sure to check out the guided lesson to catch them all. One in particular that I want to mention for you, though, is heat. You've actually denatured proteins before in your life if you've ever fried an egg. So when you first crack an egg into the pan or into a bowl, it's clear, but as we add heat, we change the 3D shape of the proteins inside the egg whites. And when they change their 3D shape, they stop being clear and they start being white. So denaturing a protein is what we see happening with eggs. You can't unfry an egg. It has permanently lost its clearness, its shape. Heat, one of the ways that I can denature a protein. I can also mess with my pH, so I could make something really acidic or really basic. Yeah, Lisa mentioned radiation. I could use radiation to damage things. Yeah, so there are a lot of different things that mess with our proteins. If we change their 3D shape and they can't fix themselves, they are denatured. Hey, not a trick question. If a protein is denatured, do you think it can still do its job? Can denatured proteins still do their jobs? No, yeah. Denatured proteins cannot do their jobs. That is why they are problematic. That, as well as this, sometimes a denatured protein has the ability to denature other proteins. So this is where we start getting into diseases. When a protein that is denatured, that's the wrong shape, starts going after other proteins that are not denatured. So here's an example of a couple of proteins that we see inside your neurons. These are called prion proteins. This is the normal shape for my protein. Hey, when I'm looking at this normal shape for my protein, do we see a lot of alpha helices or beta sheets? What does this seem to have a lot of inside of it? You can just give me A or B. Do we see a lot of alpha helices, A, or beta sheets, B? Yeah, we're absolutely right. There's lots of little alpha helices in here, little A twisties that I see. This is the normal protein, the protein that can do its job. If we go next door, we're looking at the prion protein that's been denatured. Now, when I look at the denatured protein, do I see a lot of alpha helices or beta sheets? A or B in my denatured protein? What do we see? Exactly. Several of us have completely identified it. These are all beta sheets. So here's how this goes down in your body. We've got our normal protein over here with all its nice, pretty alpha helices. Hey, this protein, by the way, we made it using directions in our RNA. We made the protein. It's got these twisty, curly Q, beautiful things on it. So here's my protein chilling out existing. Well, one day it's floating around in your neuron, and the denatured protein comes over and says, hey, you know, you'd look really good with some beta sheets. And my protein's like, well, you're probably right. I'll try it. And all of a sudden, we go from one denatured protein and one regular protein to two denatured proteins, which, remember, you told me if I'm denatured, I can't do my job. So now I've got two proteins that can't do their job. But more importantly, that protein I just gave beta sheets is going to float around inside the neuron and say, hey, you would look really good with beta sheets. Why don't you let me denature you? And it does. And the cycle continues and continues. We keep building all of these non-functional denatured proteins until it's actually toxic to the cell. It gets toxic for a couple of reasons. Number one, I'm not doing my normal job. Number two, I've got all this junk inside of me that can't do anything. So when we talk about neural disorders that come from prion proteins, what actually ends up happening is the cells themselves get so full of non-functional proteins that they die. So in the disease that you're going to be learning about this week in your application activity called Creutzfeldt-Jakob's disease, when you look at slices of the brain from people who have this disease, they literally have holes in places in their brain where there used to be neurons. But there aren't neurons there anymore because those neurons were killed by all the proteins that are formed incorrectly. Creutzfeldt-Jakob's disease, an example of a disease that's a problem with protein folding. Another one that's a problem with protein folding, when we talk about beta amyloid plaques. If you ever heard about that with Alzheimer's disease, beta amyloid plaques, they've got a whole bunch of beta sheets instead of the normal functioning shapes. So consider, as we get ready to transition into our group work, just how important it is that my proteins have the correct structure. We've got to be correct at the baseline. We've got to be correct at primary structure, the right order, for us to be able to get the right secondary structure, for us to be able to get the right tertiary structure. And if we're going up to quaternary structure, where we put more of them together, we got to get all those levels right for a protein to be able to do its job. Before I send us into groups, I want to give us a chance to ask any questions that we've come up with. So let's put our questions here in the chat. Yeah, so Alyssa asked a clarifying question about our pictures here. Yeah, the one with the alpha helices primarily, this is my normal protein. My denatured protein is the one over here that has the beta sheets. In general, Cassidy's totally right. We have alpha helices and beta sheets in our proteins all over the place. It's just with the particular protein that we're seeing an example of here on the slide, if we have only beta sheets and none of those alpha helices, we're in trouble. We need to have a mixture of alpha helices and beta sheets for this particular protein and for most of our proteins in general. So it's not like beta sheets are always bad, but they're bad when they're getting rid of where there should have been alpha helices. So good clarifying question on that. Any other clarifying questions that we have? Ooh. screen going a little crazy. Apologies. So the picture that I showed you earlier with the yellow and blue, that's a denatured protein. I suspect you're talking about this one up here. Okay, this protein is not denatured. This protein has its correct 3D shape. So it really varies from protein to protein what they're supposed to look like. This particular protein is supposed to look like this. So there are some proteins in your body that are just beta sheets. That's their normal shape. There are some proteins in your body that are just alpha helices. It's not a problem for them to exist as one or the other, but where you get into a problem is if they're supposed to be one and they're the other one, then we're in trouble. If they're in the shape that they're supposed to be, whatever that shape is, totally fine. It's when they start being the wrong shape that we're in trouble. So when they start switching from alpha to beta and vice versa, that's when we have the problem. Yeah, good clarifying question. All right, if there are no other questions, I'm going to end the recording, and then I'm going to put a link to our group activity, which is the last thing we're going to do together today. So let me go ahead and stop that recording, and we'll chat about the group work.